Hi, my name is Jessica Redline, and this is the Micro 225 lab number 9 at LAPU. It is 8/21/2020, and this is the results video. I'm going to be discussing, excuse me, my throat is hurting, the salt tolerance testing for lab 9, which we did earlier in the week. I just wanted to kind of combine the two results videos, so I wasn't doing two separate ones. So for these tubes, I saw that for the most part, the S. epidermidis in the saline tubes maintained a more clear, well, semi-clear broth, but they had more, a moderate amount of turbidity. So they were like kind of, there was either turbidity noted at the top or the bottom of the tube, while the S. cerevisiae, it was still clear, but it did have less turbidity with my experiment results. Most of them were noted at the bottom of the tube with the saline. So I thought that was interesting, and I know a lot of factors could take place with the temperature and the environmental factors of the way that the results came out. It was a little bit hard to distinguish the differences between the two, but I do think more growth happened with the sorry, I'm reading, with the S. epidermidis. Yeah, more growth happened with the S. epidermidis rather than the yeast one, in my opinion, based on my results. So I labeled my S. cerevisiae to be halo tolerant, which is meaning that it has the ability to grow in salt concentrations, but it does not require it for growth. Since I didn't see that much growth going on, I thought it fit the description as being halo tolerant. And the S. epidermidis, to me, seemed to be a halophile, which is an organism that lives in highly saline environments and require the saline to survive. And I think it actually like thrived in that environment. As far as the S. epidermidis, it seemed to have much more growth and show turbidity than the yeast did. So that was based on my results for the salt tolerance test. I can show you some examples of that. So let me see where I have my. So like this is the 15% saline, the S. cerevisiae. It's mostly clear, but you can see the turbidity noted at the bottom. The 15 in the S. epidermidis, it's for the most part clear, but there's turbidity noted like throughout the tube. Very cloudiness going on here, all throughout the tube, down here on this region, and then a big like something growth going on right here. And for the most part, that was with most of the tubes. I think a little bit more growth happened with the higher saline content for the S. epidermidis. But going on to the second portion of lab 9, this was a pH tolerance one, pH testing for the broths. And these have incubated for 24 hours, a little bit more, and they are ready to be viewed. So I have them here. I'm gonna start with S. epidermidis 5. This is pH 5, very clear. So, and it says that if it's clear broth, then it is a negative test result. So that would be negative. This is the S. epidermidis pH 7. It appears to be clear to me as well. And I didn't add any drops to this one because it was already at its neutral 7 base. This is the 9 for S. epidermidis. There is something going on top, a little bit of cloudiness and growth happening. I don't know if you can see it. I don't think you can, but it's like a film on top that's a lot thicker. Going on to S. cerevisiae 5, pH 5. This one maintained a clear result, negative result. This one is the pH 7. This shows some turbidity. You see that cloudiness at the bottom. It's a little bit cloudy throughout the tube, but I mean, it's still pretty clear, but it is mostly cloudy at the bottom. And this is the S. cerevisiae 9. So there's that. So I thought this experiment was interesting. It's a little bit confusing to see like what a positive result means. So I was doing from the reading and doing research, yeast tends to be more acidic. So the S. cerevisiae usually thrives in like 4.5, 5, but it says that it can survive in 7. So I don't know if the 7, I mean the 9 for the S. cerevisiae, it is a little bit cloudy and on the bottom. So I'm wondering if the negative result, which was the 5, is because that's the environment it does well in. So it's a little bit confusing with what differentiates like the positive and the negative. Like, does it grow better in the positive? Does it grow better in the negative? But for the S. epidermidis, I think it was more visible in definitely the pH level 9, which is more of a alkaline base, which would be positive. But doing research on S. epidermidis, S. epidermidis is usually found on human skin. And the normal pH value for skin is usually 4.5 to 5, more on the acidic side. But, I mean, in some cases it can survive on 7. I'm not sure why it made a positive result in the 9, but maybe that's just showing the environment that it doesn't do well in. But to my understanding, S. epidermidis mostly lives in, like, on our bodies in sweaty areas. So the armpits or like abdominal folds and whatnot. And usually sweat is more acidic. So the pH level ranges from like 4.5 to 5 or 6 or whatever. But then again, it can be different depending on the person's pH balance of their skin and all of that. So I thought this was a really interesting experiment to see how the cultures reacted to the different pH levels. But I definitely do want to read up more on how to properly like read the results and find out like which culture, like which pH level it actually truly does thrive in and which one it doesn't do well in. So I will do more reading on that, but overall I think it was a really good experiment and had pretty good results.